So today I'm going to tell you a little bit about the work that we've done in my lab over the past few years. Two studies that were published, and I'm going to allude to a couple of studies that were published as well without showing you data, and then tell you a little bit about our current efforts to look at aging in mice. Aging in mice is a really smart thing to do because the lifespan of a mouse is about half of the lifespan of a graduate student or a postdoc in the lab, so it's actually tractable. So first I'm going to talk about the intrinsic functional connectivity, or our attempt to actually validate that we can use mice to look at spontaneous or resting state functional connectivity. Then how we elaborated on that and looking at individual variability by doing dense sampling of these mice, and also looking at their behavior and looking at brain behavior correlations, and finally functional integrity in aging, and how we are hoping to use this approach in mice, essentially fMRI or whole brain mapping, to actually go after the biology of aging and determinants such as social factors, social isolation, genetic diversity, and different factors that confer either resilience or susceptibility in aging. So I would like to kind of mention the contributors. I'm going to focus mostly on the work that Eyal Bergman did in the lab, and allude to work that Daniela, Jad, Ben, and Amir did, and also the aging is a collaboration with Gagan Wig at UT Dallas and his graduate student Ezra. We moved a couple of years ago to Columbia University, and Amir and Daniela has been very kind of instrumental in actually setting the lab at Columbia, and we also have new members that joined the lab. The lab now is doing a lot of task-based fMRI in mice, which we are hoping will complement the resting state data that we acquired in the previous decade. Okay, so probing brain function. In the lab, we are using awake mouse imaging. So all of our mice are awake in the scanner. The way we kind of attain that goal is by essentially adopting the approaches that were developed for two-photon microscopy, implanting a head post on the mouse and acclimating it to be in a head-fixed position over a number of days, where the animal understands that it's not going to get killed, not going to suffer beyond just being in a head-fixed position. We actually kind of the setup that you saw in the previous slide evolved to be an open setup where we can actually provide a lot of the task stimulation and record the activity of the animal. So now we have videography on all of our animals when they are also in the scanner passively, but we can also engage them in tasks, and we are using all of the tools that are available to online, kind of in real time, see what they are doing and provide them stimulus responses or rewards as necessary. So what you see here on the left is essentially a mouse with a camera right in front of it. This is the single element loop coil that we use to image the entire brain, and then kind of this little mirror here gives us the view that you see here, where you can see the animal both from the front and from the side, and you can see the tongue protruding from the side. So the first thing that Eyal did kind of in his attempt to convince ourselves that resting state is something you can do in the mice is essentially scan a number of animals over many, many days. In this case, we reached almost 10 days per animal, so about 50 minutes on each day. Mostly consecutive days, and asking if we are doing the basic kind of analysis of a resting state, essentially seed-based connectivity, do we get maps that are equivalent to the ones that you would get in humans? Here you see a kind of a reconstructed surface of the mouse brain with blue and red dots, just separating them into kind of primary sensory and sometimes secondary, and then association regions, and then essentially, this is just one example of a primary somatosensory seed map that is marked here by a little asterisk, and we're getting a pattern of kind of connectivity across the entire brain. So these are just spontaneous fluctuations of the fMRI or oxygenation in the blood. And what we can do is we can compare that to a ground truth track tracing from the Allen Institute. So you take the Allen Institute it will show up again in a moment, a highly detailed connectivity map. And you can just ask for overlap between these regions. You can see that there's also like the traveling fiber bundles. So we ignore them when we just look at like target regions and ask, what is the amount of match between these different regions? So this is how a structure function overlap looks like. And this is the reproducibility across animals. In this case, it was 12 animals. So this is like a very kind of a simple way to just get an impression of how these two kind of things are matching, and we quantify that in a number of ways. Here at the top, we just did some sanity checks, overlapping, meaning overlapping with anatomy versus kind of voxels that are not overlapping with track tracing anatomy, and the expectation was that for the most part they're going to be consistent, and that's indeed the case. Just to remind you, spontaneous resting state connectivity does not obey only a single synapse distance, and the track tracing was only single synapse. And this is the kind of maybe the difference. We also did the ordered connections, so again, anatomical connectivity and how much coverage we have, and just ordering them by significance. So the biggest projections first, the least projections last, and then asking in the resting state how many kind of the ordered connections we need in order to get coverage, controlling for size. And what you can see here is that size, which is an important factor, doesn't explain everything. And on the right, you can also see that if you do a shuffle analysis, the kind of the functional connectivity is above random connectivity. The last analysis that we've done, was kind of a threshold independent approach using receiver operating characteristic, where you essentially hold the kind of track tracing information constant. So you establish a threshold for the optical imaging there, and then you vary the threshold of the functional connectivity from very conservative to very lenient, and essentially true positive versus false positive, where the expectation is that we're going to get a curve for more of the true positive, and if it's completely random, just by association. So, another interesting difference that we've seen here is the difference between association and sensory regions. So, the expectation is that sensory regions will obey more kind of hierarchical versus parallel distributions. The next thing that we've done was to look at the individual variability. In this case, we scanned the number of animals for, again, multiple days, split the data into halves. This is a whole brain connectivity of 3 example animals, showing the first half of the data on the top and the second half on the bottom, and then 3 different animals. And you can get an impression of how much each animal kind of matches itself versus the other animals. And you can see that even just by eyeballing these kind of matrices, each animal is more similar to itself than the others, and there's also similarity across different animals. Again, we quantify that by just correlating on the diagonal the animal to itself, and below the diagonal each animal to all of the other animals. And if you quantify that, you can actually see that if you match all of the animals to the average group, they are below the network similarity from the animal to itself. And this is just to mention that we followed here work that's been done in humans. So we actually kind of went step by step on work that was done by Katerina Graton, Emily Finn, showing that there's this match. The last thing that we've done also to look at the kind of match of brain behavior is to look at the kind of simple task, the rotor-rod task, which is kind of a motor coordination, and asking whether we can explain brain behavior using this type of task. So this is the latency to fall of each of these animals. Each gray line is an individual animal, and you can see that the animals on average learn. There's a lot of variability. It's a noisy task, but if you use a connectome predictive modeling, so essentially using the whole brain data, doing a leave one out analysis approach, to try and explain the behavior, we can identify a set of voxels in the brain that can explain the behavior. So this is the observed mean latency to fall versus the predicted latency to fall. We can even use these both positive and negative contributions to try and pinpoint where in the brain this kind of explanation is coming from. So it's not all systems. So this is kind of looking at sets of networks. And here is the kind of the main contributors to the positive kind of prediction of the responses. So sensory connectivity, either within or between networks. And this is, for example, a negative contribution to this task. So association sensory regions showing high correlation will predict worse performance of each animal. So I hope that kind of this really brief kind of breezing through these sets of results kind of convince you that we are able to replicate kind of evidence shown in humans. And what we've now are doing, so we actually done a number of studies looking at developmental disorders and using also rescue using therapeutics. And now we are looking at aging. So here we use the measure that Gagan Wig used in humans to look at within versus between connectivity of separate networks in the brain, looking at segregation as a measure of aging. Gagan in a set of works has shown that there are these measures of network segregation. So here you can see intact network segregation in a single individual that is maintained across about 6 years. And here you can see the falling apart of this network segregation. And what Gagan has shown is that this measure is a predictor of AD pathology, APOE status, and overall cognitive health. And what we've done now in the mice is use the same type of kind of a measure to look at we kind of fine-tune the brain's parcellation coming from the Allen Institute, showing that we can obtain this measure of communities, which we kind of then can interrogate for system segregation, and show that between 3 and 12 months of age in the mouse, we see a decrement in this segregation, but we can also do that in a longitudinal manner in individual animals, showing that essentially these animals also show network segregation decreases as we are seeing in humans. And this is just showing kind of over a number of cohorts. We have by now, since we've been scanning for many years, we have more than 200 animals with resting state data, and about a third of them with different ages, and we are seeing that this is following the trajectory that we are seeing in humans. So here I'm kind of putting kind of the conclusions in general, but also again highlighting that what this kind of approach gives us is the ability to look at the determinants of healthy versus unhealthy aging. We can look at diverse, genetically diverse mice that recapitulate genetic diversity in humans, looking at what type of factors confer resilience versus susceptibility. We can also use social isolation approaches to look at kind of what kind of puts an animal on a kind of a bad trajectory for aging. Again, thanking all of the different collaborators and financial support, and thank you.
Thank you, Itamar. Does anyone have a very short and quick question to Itamar? Going once, going twice, sold. Yeah.
Ido.
Take everyone's time to the last one. Just a quick question. So, great talk, thank you. You showed this sort of a fingerprint on mice and rats. Sorry, it's mice or rats?
Mice.
Mice. Amazing. I just wanted to ask, are they genetically identical, the animals?
So genetically they're identical, but still we are seeing differences in their behavior. And that's something that has been kind of a...
Because give a great opportunity to ask about the origin of fingerprinting, right? Like
exactly.
Nature versus nature, actually. Yeah. So let's discuss further on later on, but great talk, so thank you very much.
Yeah. And also Alex's predictions are something we can test on these animals.
Definitely.